Good evening, students. I would invite Dardi ma'am to start the session.
Good evening, students. So let's start the session. Okay, hope my screen is visible.
Yes,
So,
ma'am, your screen is visible.
okay, thank you.
Yeah.
So today we'll discuss the last division, that is the angiosperms. Earlier, algae, bryophytes, pteridophytes, and gymnosperm is completed. So today we will discuss the last one, that is the angiosperms. And after this, we will, study everything about these angiosperms only, like sexual reproduction in flowering plants and all. So this is very important topic, that is the angiosperms, and which is the most advanced plant group. Okay, so angiosperms. So what is angiosperms? So these are also known as the flowering plants and are the largest and most diverse group of plants on earth. So earlier, till gymnosperm, it is not flowering. Like pteridophytes and the gymnosperms, they are vascular, but these angiosperms are flowering plants. Means they produce both fruit and flower. They are of course vascular also, and these are the largest and they have the most diverse group of plants on earth. They are distinguished by several unique features that set them apart from other plants like gymnosperms, conifers, and fern. So basically, few things are similar with the gymnosperms, and these angiosperms also has some unique unique characteristics, so that you can differentiate the gymnosperms and the angiosperms. So let's study the general characteristic of these angiosperms. So number one is the flowers. So angiosperms are characterized by their production of flowers, which are the reproductive structure of this plant. So basically, the reproduction takes place through these flowers. So this flower is the main part of the plant, and it is also known as the reproductive structure of these plants. Flower facilitate the process of pollination, which is the transfer of pollen from male to female plant part. So the flowers, this from these flowers, the pollination process also take place. And what is pollination? That is the transfer of male gamete, the pollen, to the female gamete, that is the female. That is called pollination. So this is crucial for the fertilization and subsequent product of seed. Production of seeds. So basically, in angiosperms, you will see that flowers are very much showy. They are very much attractive. They are brightly colored. Why so? So that along with wind and water, there are also animal pollinators or insect pollinators. Okay, like butterflies, moths. So everything will come and do the pollination process. So these are, very much attractive. The flowers are very much attractive. You have seen the hibiscus, then roses, sunflowers. So very much darkly, brightly colored and very beautiful flowers, to attract the pollination process. Okay, so these are the main reproductive structure of these plants. Next is the fruits. So after fertilization, angiosperm produce fruit. But you see in gymnosperm, gymnosperm lack the fruit. Okay, this is again a differentiating factor of the gymnosperm and the angiosperm. So in angiosperm, after fertilization, it produces fruits. A fruit is a mature ovary that contain the seeds. So basically, the fruit is nothing. It is the ovary itself, but it is matured one. And this ovary means this fruit contains the seed. So what is fruit? Fruit is the matured ovary, okay, or the modification of the ovary. It is called matured ovary that contains the seed. Fruits help protect developing seed and can aid in their dispersal by attracting animals or through other mechanism like wind or water. So basically, this fruit, what is the purpose of this fruit? This protect the developing seeds. It gives protection to the developing seeds and can aid in the dispersal and help also in the dispersal to attract the animals and through other mechanisms like wind or water. So this is the fruit. Okay, in earlier, like gymnosperms, the seeds, they are basically exposed in the cone or other structures, but here it is covered within the fruit, means the matured ovary. Don't forget, fruit is the matured ovary and it contains the seeds. Next is seeds with endosperm. So an angiosperm seed typically contain endosperm, a tissue that provides nourishment to the developing embryo. This is a significant feature because it supports the seedling's initial growth after germination. So basically this endosperm is also very unique characteristic of the angiosperm plants. Okay, here you will find the endosperm. So basically what is this endosperm? This is also a product like the zygote. This endosperm is also a product of the fertilization. Means in angiosperm, like in earlier we have seen, fertilization takes place only one time. That is, the male gamete will fuse with the female egg and the zygote will produce. But in the angiosperm part, we will see that there are two fertilizations. One, the male gamete, the sperm will fertilize with the egg. Another, one more sperm will fertilize the two polar nuclei. That is a double fertilization, and one will produce the zygote, but the fertilization between the male gamete and the two polar nuclei will give results into endosperm. So basically this endosperm is a tissue that provides nourishment to the developing embryo. Means the zygote will definitely grow into a embryo, and this endosperm will give the nourishment and the nutrients to this embryo, and it supports the seedling initially in the growth. So basically this role is to give the nourishment, the endosperm rule. Next is the double fertilization. I have told a distinctive process in angiosperm is double fertilization. During this process, one sperm cells fertilize the egg cell to form the zygote, while another sperm cell fuses with two other cells to form the triploid endosperm. This results in both the embryo and the nourishing endosperm. So this is the process of double fertilization, where one sperm cell fertilizes the egg cell to form the zygote, while the another sperm cells fuses with two other cells, that is the polar nuclei, to form the triploid endosperm. So don't worry, our next chapter is the reproduction in flowering plants only. So there we will discuss more deeply about this double fertilization, what is polar nuclei, what is, how it will from, what is chalajal, micropyle, and so we will discuss everything briefly. Here just you remember that in angiosperm there is a phenomenon takes place that is called as double fertilization, and as a result endosperm is formed with the seed. Okay, this is the key feature of this angiosperm. Next is xylem structure. Angiosperms have a more advanced xylem structure compared to gymnosperm. So gymnosperm has also xylem structure, but in endosperm it is more advanced from, because it is the most advanced plant group. The xylem tissue includes vessel elements, which are specialized cells that aid in the efficient transport of water. This is a key adaptation for their diverse habitats. So in gymnosperms, yesterday we have discussed that their vessel elements are absent. There you will only find the tracheids. But here in the angiosperms, you will also see the vessel elements that are more specialized cells. Their work is to transport the of water efficiently, more productively. So angiosperms, they have an advanced xylem structure, and it contains the vessel elements, while gymnosperms lack vessel elements, and these are specialized cells that aid in the efficient transport of water. And this is the most key adaptation of their diverse habitats. Next is variety of growth forms. So angiosperms exhibit a wide range of growth forms, including trees, shrubs, herbs, and vines. This diversity allows them to occupy many different ecological niches. So they exhibit in various forms like trees, shrubs, herbs, and vines. Next is broadleaf types. So we are studying the general characteristic of the angiosperms. So broadleaf types. So the leaves of angiosperms can vary greatly in shape and size, from broad and flat to needle-like. This diversity in leaf morphology helps them to adapt to various environmental conditions. So you will see leaf types are variation from species to species, the shape and size. Somewhere it will be small, somewhere it will be large, from broad, somewhere it will be broad, somewhere it will be flat, to the also you will see the needle like leaf. So there is a very huge diversity in the leaf morphology, and it helps them to adapt in various environmental conditions. Next is symbiotic relationship. Many angiosperms form symbiotic relationship with other organisms such as mycorrhizal fungi, which assist in nutrient uptake, and various pollinators which aid in reproduction. So, in angiosperms also you will see this. The higher plants, higher trees, they produce symbiotic relationship with fungi, and the fungi will also get the nutrients and all, and also in return they will get the pollinators. So this is symbiotic relationship. So this is all about the general characteristic of the angiosperms. Now we will discuss the cell structure of the angiosperm. So what is the cell structure of angiosperm? The cell structure of angiosperm flowering plant is characteristic by several key features that reflect their complex and highly specialized function. So basically, more advanced form this is. So the cell structure is also advanced. It is a complex one. Okay. Number one is the cell wall. So there are two cell wall, primary cell wall and secondary cell wall. So the primary cell wall, it is made up of cellulose. Then apart from cellulose, you will also find hemicellulose and pectin. It provides structural support and define the cell shape. So this primary cell wall, it is mainly composed of the cellulose. Okay, like other plants group. But apart from cellulose, you will find hemicellulose and pectin also. And this primary cell wall, what does they do? They provide structural support, and it also gives the shape of the tree, shape of the plant. It defines the cell shape. Okay. Next is secondary cell wall. In some cells, especially in xylem vessels and fibers, a secondary cell wall is deposited inside the primary wall. This secondary wall contains additional layers of cellulose and lignin, which provides extra strength and rigidity. So apart from the primary cell wall, so you will also find secondary cell wall in xylem vessels and fibers. Means a more layers of cellulose and lignin will be deposited, and a secondary cell wall is formed. And what it do, it gives extra strength and rigidity. And also, sometimes in some matured and old tree you will find tertiary cell wall also. Okay, apart from primary and secondary, tertiary cell wall also fine. Number two is the cell membrane. So that is plasma membrane. So this is again same like the pteridophytes and bryophytes. This is a phospholipid bilayer which embedded proteins. So there are two lipid bilayer with where the proteins are embedded, that regulates the movement of substances in and out of the cell. So it basically helps in the movement of the some substances inside the cell and outside the cell. It plays a crucial role in maintaining cellular homeostasis and facilitating communication between cells. So it provides optimum condition to the cells, and it facilitates communication between the cells. And basically this plasma membrane, they are very much selective to the materials, membrane selectivity. So all elements, all substances can't go inside or can't expel outside. So it has some selectivity. It has some receptors, it has some signaling proteins, and like that, which whatever substances is needed inside, it will allow only that substances inside. So plasma membrane is highly selective. It will not allow everything to go inside. Okay, you come, you come, you come, not like that. It is very much selective to the substances. This is the cell membrane. This is the plasma membrane. Next is the cytoplasm. So in cytoplasm, there is cytosol, the liquid portion of the cytoplasm where metabolic processes occur. So in cytosol, all the metabolic processes take place. Next is the organelles, various specialized structures suspended in the cytosol, including mitochondria, chloroplast, endoplasmic reticulum, and Golgi apparatus. So, in the cytosol, basically this is a gel-like matrix. There you will find all the organelles, from mitochondria to the Golgi apparatus. So, all the organelles are, present within the cytoplasm in the gel-like matrix. Next is nucleus. So nucleus is the main part of the cell, because it contains the DNA. it contains the inheritary material. So nucleus, it is composed of nuclear envelope and nucleolus and chromatin. So nuclear envelope, it is a double membrane surrounding the nucleus, which contain nuclear pores for exchange with the cytoplasm. So basically this nucleus is surrounded by the nuclear envelope. So there is a envelope, means there is a protective layer. Yeah, I mean it is a double membrane layer which surround the nucleus, and it also contains in the layer you will see some nuclear pores. I mean there are some teeny hole like things, teeny holes, teeny pores are there, and this pores is for exchange of things with the cytoplasm from nucleus inside to the outside. This nuclear pores are there, and in nuclear pores there you will find many proteins. Many nuclear proteins are there in the nuclear envelope. Next is the nucleolus. It is a dense structure within the nucleus where a ribosomal RNA synthesis occur. So in nucleolus, what is this? This is a dense structure which is found within the nucleus, and here ribosomal RNA synthesis occurs. Here you will see the synthesis of the RNA. Okay. You will see here transcription method. Next is the chromatin. So don't worry this for these terms like what is RNA synthesis, like what is protein synthesis. So we have to discuss all these things in our molecular biology, where we will discuss all DNA replication, transcription, translation, and those things are very much interesting. So we'll learn everything. So here you will just remember the terms, what's are present in the angiosperms, what are the things that is present in the angiosperm cell structure. So yeah, in the nucleus, nuclear envelope is there. That is next is the nucleolus. That is a dense structure within the nucleus where ribosomal RNA synthesis occur. So next is the chromatin. So that is DNA associated with histone proteins, which condense to form chromosomes during cell division. So there is the presence of chromatin. So that is the DNA, the main genetic material associated with some histone proteins, and it very much condenses to form chromosomes during cell division. So basically these chromosomes contain this DNA for the cell division, like the meiosis and mitosis. I have already explained that meiosis is the reduction division. Like due to this meiosis, from diploid we again get the haploid cells. Okay, and mitosis is the repetitive divisions of the cells. So nucleolus contain nuclear envelope, nucleolus, and the chromatin. Next organelle is the chloroplast. So which is responsible for photosynthesis, the main thing in the plant, greenery plant. So chloroplast contain thylakoid membrane. These are internal membranes arranged in stacks called grana, where photosynthesis occur. So the chloroplast has some thylakoid membranes, and these are internal membrane, and it is arranged in stacks. Stacks means like one above another. So these arranged in stacks, and these stacks are called as grana. Okay, and in grana the photosynthesis occurs. Okay, next is the stroma. This is the fluid filling space surrounding the thylakoids containing enzymes for the Calvin cycle. So what is stroma? What is grana? So grana is nothing. It is the thylakoid membrane arranged in stacks, means one above another. That is called grana. And stroma is the fluid filled space surrounding the thylakoids. Means this stroma will surround the thylakoids, and this is fluid, basically a fluid filling space. Okay, and the stroma contains all the enzymes for Calvin cycle, means like photorespiration cycles, and for other metabolic activities. The stroma contains all the enzymes. That is the chloroplast. So due to this chloroplast, the photosynthesis takes place, as it also contains the main pigment, that is the chlorophyll. Next is the vacuole. Next organelle is the vacuoles. So there is central vacuole, a large central organelle in plant cell that stores nutrients, waste products, and help maintain turgor pressure by exerting pressure against the cell wall. It is filled with cell shape, a mixture of water, ions, sugar, and other substances. So there is a very large central vacuole present in the plant cell, and what is the main role of this vacuole? It stores nutrients, waste products, and helps maintain the turgor pressure by exerting pressure against the cell wall. So cell wall has a pressure, so it maintains that target pressure by exerting its pressure. It is filled with shell shape, means it is the vacuole is filled with a mixture of water, ions, sugars, and other substances. And main role is to store the nutrients. That is the role of the vacuoles. Next organelle is the endoplasmic reticulum, ER. So there are two ER. One is the rough endoplasmic reticulum. One is the smooth endoplasmic reticulum. So the rough endoplasmic reticulum studded with ribosomes. It is involved in the synthesis of proteins that are either secreted from the cell, incorporated into the cell's plasma membrane, or sent to an organelle. So basically, this rough endoplasmic reticulum, its role is to synthesis of proteins. The rough endoplasmic reticulum synthesizes the proteins. The translation processes takes place in the rough endoplasmic reticulum. And it is studded, it is filled with the ribosomes. Next is the smooth endoplasmic reticulum, smooth ER. So basically it lacks ribosomes and it is involved in lipid synthesis, metabolism, and detoxification processes. So rough will synthesize the proteins and smooth ER will synthesize the lipid. Okay, this is the difference between the rough and smooth. One rough for protein and smooth for lipid. Next is the Golgi apparatus. So, cisternae, flattened membranous sacs that modify, package, and short proteins and lipids received from the ER. It is involved in the production of cell wall materials and various cellular secretion. So, this Golgi apparatus contain a structure, flattened membranous sacs, that is known as cisternae. And this cisternae, what they do? They modify, means whatever they received from the endoplasmic reticulum, the lipid and protein, they modify. This cisternae will modify it. If some modification is needed, then it will pack it, short it, and it will deliver it to some other destination wherever it is needed. Basically, this Golgi apparatus, this cisternae acts as a modification and delivery agent, like Swiggy, Zomato. Okay?
Yes, ma'am.
Yes. So, next is the ribosome. So, ribosomes, so there are ribosomal units. So, small structure composed of RNA and proteins responsible for protein synthesis. So, basically, these ribosomal units, they are responsible for protein synthesis. The process is also known as a translation. First, DNA replication will take place. Then from DNA, RNA will form, like the process of transcription. Then from RNA, the proteins will be formed. That is the process of translation. So, a ribosomal unit form is the main thing that is responsible for protein synthesis. In plant cell, ribosomes can be found both free in the cytoplasm and attached to the rough endoplasmic reticulum. So basically, these ribosomal units. So there are very big topic of these ribosomes, like what type of ribosomes are there, the division of ribosomes, the units of ribosomes. So basically, these ribosomes, they are found in two conditions. One is free in the cytoplasm without any disturbance, and next is it is also attached to the rough endoplasmic reticulum. Okay, and this rough endoplasmic reticulum is responsible for protein synthesis, and smooth is responsible for lipid synthesis. That is the ribosomes. Next is mitochondria. So this is also known as the powerhouse of the cell. Okay, this provides the energy. So it is have inner membrane folded into cristae to increase surface area for ATP production. So the inner membrane, it is vigorously folded, means from one fold to another, one fold to another. So it is vigorously folded, and the folded structure is known as cristae. Okay, and why they are folded? Why cristae is formed? To increase the surface area for ATP production. More the surface area, more ATP will form. So basically, what is this ATP? So the full form is this ATP is adenosine triphosphate. Adenosine triphosphate. So this is a molecule which provide energy for many processes within living cells. So this is sometimes this question is asked that which is known as energy currency. So ATP is known as energy currency of the cells, because it stores and transfers energy necessary for various cellular function. So the ATP, it is known as adenosine triphosphate, and it is a molecule that provides energy for many processes within living cells. Okay, and often this ATP is referred as energy currency of the cell. So, what is known as energy currency of the cell? The answer is the ATP, because it stores and transfer energy necessary for various cellular function. Okay, so this is ATP, and it is found in the mitochondria. Next is the matrix, that is the gel-like substance inside the inner membrane where the Krebs cycle takes place. So, apart from the inner membrane, there you will also find the matrix inside the inner membrane. So what does this matrix do? So this matrix, it contains some substances, means like due to these enzymes the Krebs cycle takes place. So basically, in short, we'll learn the Krebs cycle later. So this Krebs cycle is also known as a citric acid cycle or TCA cycle. Okay, and it is a part of cellular respiration. Like you human beings, plants also respire. Okay, so the cycle name is like Krebs cycle, or also known as TCA cycle. Okay, so basically it takes place in the mitochondria of the eukaryotic cells, and it has many series of chemical reactions like oxidation, acetyl CoA. So it's a very big process, a very big and complex process of this Krebs cycle. So in physiology you will learn, basically now you know that this is also known as TCA cycle, that is important for cellular respiration, and it takes place in the mitochondria. Next is peroxisomes. So peroxisomes, what are these? Organelles that contain enzymes for breaking down fatty acids and detoxifying harmful substances. So basically these peroxisomes, these are also found in the cytosol of the cytoplasm. So these are the organelles, and it contains enzymes for breaking down the large fatty acids, and it neutralizes the harmful substances. That is the role of the peroxisomes. Next part is the plasmodesmata. So you know what is plasmodesmata? They have some channels. These are very microscopic channel that transverse the cell wall of plant cells, and it allows direct exchange of water, ions, and small molecules between adjacent cells. Means between the neighboring cells. Due to this plasmodesmata channel, it is possible of direct transfer of exchange of water, ions, and small molecules. That is the role of the plasmodesmata. Apart from this, all cell organelles like mitochondria, chloroplast, peroxisomes, plasmodesmata, the nucleus. Okay, so apart from this, there are also some specialized cell present in the angiosperms. So what are these specialized cell? Number one is the xylem cell. So it includes tracheids and vessel elements. The xylem cell includes tracheids and vessel elements. So remember, vessel elements are absent in the gymnosperms. Only tracheids is present. But here, you will see, you will find tracheids and vessel elements in the xylem cells, which are specialized for the transport of water and nutrients. Vessel elements are characteristic by their perforated end wall. So end walls, they have some small pores in the vessel elements. So this is the xylem cells. Next is the phloem cells that include sieve tube elements and companion cells. So in gymnosperms, we have only find sieve cells in the phloem cells. It lacked the sieve tube and companion cells, but here in angiosperm, you will find sieve tube elements and the companion cells also, which are involved in the transport of sugars and other organic nutrients. Sieve tube animals have pores in their end walls, that is called sieve plates, to allow flow of nutrient solution. So basically, these sieve tube elements that is present in the phloem cells, they have some pores in their end walls. Means end of the wall, it has some pores that is called sieve plates, and it allow the flow of nutrient solution. It helps in the flowing of the nutrients, transportation of the nutrient solution. That is xylem cells and phloem cells. This is a thing about the growth of the primary growth, cell wall of this is the primary growth cell wall of the angiosperms, where you will see these are the phloem, this is the xylem, this is the sclerenchyma cell, and this is the I have said that here you will also find secondary growth. So due to the secondary wall, you will see the apart from primary xylem and primary phloem, there is also secondary xylem and secondary phloem. Then there is cork, bark, cortex, epidermis. So it's a very complex structure in the angiosperms, like stem. So like when we will study the anatomy, we have to study everything, like I don't know, like in practicals also, there is dissection of the stems and all, dissection of the species to see the anatomy, the internal structure, internal cells of the plant. Okay, so now we'll study the reproduction in angiosperms, which we also have to study in our next chapter very deeply. So, the reproduction of angiosperms flowering plant is a complex and highly coordinated process involving both sexual and asexual mechanism. So, the angiosperm flowering plants reproduction that also contain both sexual and asexual mechanism. So, sexual reproduction in angiosperms involve the fusion of male gametes and female gametes to produce seeds. This process typically includes several stages. So, sexual reproduction is, of course, the fusion of male and female gamete to produce the seeds, zygote, then seeds, and it involves several stages. So, number one is flower structure and pollination. So, it has some complex flower structure. So, basically, the flower is the reproductive structure of angiosperms. Means the flower is the main reproductive structure. In flower, you will find the male part and the female reproductive part, which contains the male part that is called the stamen and the female part that is called the pistil. So, male reproductive part is known as stamens and female reproductive part is known as pistils. So, male is also termed as androecium and female is termed as gynoecium. Remember, in angiosperm, there two terms are used: androecium for male, that is the stamens, and gynoecium for the female, that is the pistils. So, stamel consists of an anther where pollen is produced and a filament. So, stamen has two parts. One is anther. In the anther, pollen, the male gamete will be produced and a filament. While pistil consists of a stigma where pollen lends a style, a tube containing the stigma to the ovary, and an ovary which contains the ovules. So, in female gynoecium, the pistil consists of a stigma, and in the stigma, the male gamete pollen will lend. Then you will find a style, that is a tube-like structure, and the style will connect the stigma to the ovary. And ovary is the basal part, bottom part, which contain the ovules. Let me show you the okay, this is a typical flower. So you will see that the yellow part is the petals. Then the one green part, long green part is the gynoecium. Okay. In the gynoecium, the upper part is the where I have marked it. The upper part is the stigma, then style, and bottom part is the ovary. And the yellow one, which I have marked as the yellow one, that is the anther, the male reproductive structure. You will see here more. This is the androecium, the side 2 one, that is the androecium. The androecium, you can see in this right-sided structure that the stamen, it contains microsporangia, pollen grains in the anther, and this is the long is the filament in the right structure. In this one, you will see this is the androecium, stamens, pollen grain is there inside, microsporangia, and it is the filament. And this is the left one is the gynoecium, means the carpels, where upper is the stigma, there is a style, and next is the ovary, ovules, where megasporangia will produce. So this is a typical structure of the gynoecium and androecium in the flower. Okay. So, okay. So this is about the flower structure. Next will come pollination. So the transfer of pollen from the anther of one flower to the stigma of another flower. Pollination can be facilitated by various agents, including insects, which is also known as entomophily, wind, which is known as anemophily, and water, which is known as hydrophily, and animals. So pollination is basically transfer of pollen from anther to the stigma of another flower. Means from one flower to the another flower. So this pollination can be, proceed through various agents, including insects. So that is insect pollination is known as entomophily, wind is anemophily, water is hydrophily, and there is animals. So next is fertilization. So after pollination takes place, pollen germination will takes place. Upon reaching a compatible stigma, pollen grain germinate and produce a pollen tube that grow down the style to the ovary. So look, there are many flowers, in our surroundings. Suppose there is hibiscus, there is rose, there is something more, sunflower. So like that, like suppose there is a female part, female pistil is there in a flower. Okay, so in that female pistil, never think that only the male gametes, means the pollen of that flower will come only. There are many pollens of different flowers can land in that pistil. In that pistil, many from the different flowers, different pollens can land. But all that pollens germinate. The question is that all the pollens are able to germinate in their stigma. The answer is no. All pollens can't germinate. In the pollen, like in the stigma, in the pistil, in the stigma, there are some there are some enzymes. There are some receptor enzymes. So these receptor enzymes recognize that which pollen is compatible for this flower, means which pollen is fit for this flower. So it will allow only that pollen to germinate in that flower. Okay, it's like a lock and key. All key can't open the lock. Okay, all keys can't open the lock. So there is specific keys for specific locks. So this is the main mechanism, that all pollens from all the flowers can't germinate. Okay, so there are some specific enzymes present in the stigma, so that enzymes recognizes when the compatible pollens reaches that the surface. Okay, and then the germination process takes place. Okay, so upon reaching a compatible stigma, pollen grains germinate and produce pollen tube that grows down the style to the ovary. So after recognition, then the pollen will germinate and produces a pollen tube, and this pollen tube will grow down the style to the ovary. Next is double fertilization. This unique process involves two fertilization events. One sperm cell fuses with the egg cell to form a diploid zygote. Another sperm cell fuses with two other cells, that is the polar nuclei, in the ovule to form a triploid endosperm, which nourishes the developing embryo. So this is double fertilization, means two-time fertilization takes place. One time, one sperm cell will fuse the female gamete egg cell, and it will produce a zygote twice. And next, another fertilization will takes place where the another sperm cell will fuses with two other cells, that is the two polar nuclei in the ovule to form a triploid endosperm. This is not neither haploid nor diploid, it is triploid, that is trisend. Okay, it is trisend, which nourishes the developing embryo. This endosperm role is to give nourishment to the developing embryo. That is a double fertilization. Next is seed and fruit development. So after double fertilization, seed will form. After fertilization, the ovule develop into a seed containing embryo, endosperm, a seed coat. So the ovule will develop into seed, and the seed contains the embryo, the endosperm, and a seed coat. So embryo is the main thing, and the sperm will give nourishment to the embryo, and the seed coat will give the protection. Next is fruit formation. The ovary matures into a fruit which protects the seeds and aids in their dispersal. So after some time, the ovary will also mature into a fruit, and this fruit will protect the seeds and also helps in their dispersal. Next is seed dispersal. Method: seeds are dispersed through various mechanisms, including wind, water, animals, and mechanical means. This helps in spreading the plant species over a larger area. Then once matured enough, it will disperse in the air, water, or some animal will take it. So like that. Okay, so this is the sexual reproduction from pollination to the seed dispersal. So next is germination. So there are some conditions for germination. For germinating seeds typically require suitable environmental conditions, including water, temperature, and sometimes light. So, after dispersal, for germination of a seed into a new plant, it requires some conditions like water, temperature should be minimum, and sometimes light is also important. So process is that upon germination, the seed absorbs water, swells, and the seed coat breaks. The embryo start to grow and the radical root emerges first, allow followed by the shoot, stem, and leaves. So once the seed, after dispersal, once the seed will, fall into some suitable condition, it starts germinating. The seed will absorb the water, and due to that water absorption, it will swells. You have seen sometimes moong, and you have seen, like things we eat, like moong and all, it swells up when it takes the water. Plasmolysis takes place. It will swell up, and the due to this swelling, the seed coat, the protective layer will break down. Then the embryo will start to grow, and first a radicle, a root will emerge out. A radicle, a root will form. After the root, shoot, stem, and leaves will also form. So this is the germination process of the new plant. First radicle, then shoot and leaves. So, okay, this I have already mentioned. Okay, this is the life cycle of the sexual reproduction. You can see this is the microgamete. Sperm mitosis will take place. Microsporophyte is there. Again, meiosis will take place, and due to meiosis, the gametes are haploid. That is mega. Then next, the right one is the megagametophyte. That is the female. That is the female one. Egg will be there. Then fusion will be takes place. Again, mitosis. Then megasporangium. Then meiosis. Again, megaspore will form. So this is the life cycle of the angiosperms. Next, after sexual reproduction, there is asexual reproduction. So, asexual reproduction allows angiosperm to reproduce without the need for seeds for pollination, producing genetically identical offspring. So, asexual reproduction, it produces without the need for fertilization or the pollination. So, modes are vegetative propagation. We have already learned these terms earlier many times. So, vegetative propagation, that is the runners or stolons. So, these are horizontal stems that grow along the soil surface and produce new plants at the nodes. So, example is strawberries. Next is the rhizomes. So, these are also underground stems that grow horizontally and produce new shoots. Example is ginger. Next is tubers, that is the solen underground stems that store nutrients and can produce new plants. That is the potatoes. That is bulbs, underground storage organs composed of fleshy leaves or scales that can produce new plants. That is the onions. So we have already learned vegetative propagation many times. That's why I'm not explaining more and more again. So these are the terms: runners, rhizomes, tubers, and bulbs. Next is clonal reproduction. So our last class, we have discussed about the cuttings. Like example, I have given of rose, like some pieces of leaves or roots is cut from the parent plant, and that can grow into new plants. We will plant it in another soil and it will grow into new plants. And sometimes some rooting hormones is also given. Next is grafting. This is a technique where tissue from one plant are jointed with those of another to grow together as a single plant. This is commonly used in agriculture and horticulture. So what is grafting? Suppose there is two plants, plant A and plant B. Suppose plant A's fruit is very much sweet, and plant B is a long-lived plant. Okay. So what we will do? We will take one part from plant A and one part from plant B. So we will join that together. Of course, there are some hormones which help in that process, and it will join as a single plant. So basically, we will get the desired characters in that single plant of the two plants. That is called grafting. That is apomixis. It is a very new term, apomixis, in the angiosperms. So apomictic seeds are what? Some plants produce seeds without fertilization. So basically, for production of seeds, fertilization is necessary. But there is some exceptions that some plants produce seeds without fertilization, resulting in offspring that are genetically identical to the parent plant. This can occur through various mechanisms, including the development of seeds from the ovule without fertilization or from the diploid cell of the ovary. So this process is called apomixis, where seeds will develop but without fertilization. This is called apomixis. So each method of reproduction provides angiosperms with different advantages for survival and adaptation. Sexual reproduction promotes genetic diversity, while asexual reproduction allowed for rapid and efficient propagation of successful genotypes. So, this sexual reproduction will promote genetic diversity. We'll get, variety, while asexual reproduction will restore identical plants, and it is rapid in process. So angiosperm, like all land plants, it also exhibit alternation of generations. So basically, it is a fundamental aspect of the life cycle. So this process involves alternating between two distinct phases, the sporophytic generation and the gametophytic generation. So here also you will see alternation of generation. Sporophytic generation will be there and gametophytic generation will be also there. So sporophytic generation, it is a diploid phase, of course, twice N. So the sporophyte is the dominant diploid twice N phase of the angiosperm life cycle. It is the phase that most people recognize as the plant, including its roots, stems, and flowers. So the plant itself is the sporophytic generation. So flower formation will take place within the sporophyte. Flowers are produced, which are reproductive structure containing both male and female. And this flower will contain the male structure and the female structure. The stamen is the male, which consists of an anther, which will produce pollen and a filament. I have already shown you the diagram. And the female structure, the pistil, which includes the stigma, where pollen will be land, pollen reception, style that connects stigma to ovary, and the ovary contains ovules. So this is the diploid phase. The production of flowers and the plants. So this is the diploid phase. So meiosis and spore formation. Inside the flower, specialized cells undergo meiosis, that is a reduction division, to produce haploid spores. So these are microspores. Micro means, small. This is the produced in the anther of the stamen, and this develop into pollen grains, male gametophytes. So of course, meiosis takes place. So these are again haploids. Okay. Next is the megaspore. So it produced in the ovules, which in the ovary, this develop into embryo sex female gametophyte. So megaspore will give rise to ovules, and microspore, micro means small, mega means large. Microspore will give rise to the pollen grains. Next is the gametophytic generation, that is the haploid phase. The gametophyte generation is a haploid N phase that is typically reduced in angiosperm. It is short-lived, okay, compared to other plant groups. It consists of the pollen grains, male gametophyte, and the embryo sac, female gametophyte. Means production of the gametes is known as the gametophytic stage, which is, of course, haploid. Next is pollen grain development. So pollen formation, microspore undergo mitosis to form pollen grains. So microspore will go repetitive division and pollen grains are formed. So each containing 2 or 3 cells. These cells include the generative cell, which will divide to form 2 sperm cell, and the tube cell, which will form the pollen tube. So basically the pollen grain, it contains 2 or 3 cells. So basically the pollen grain contains 2 cells. One cell is known as the generative cell and the other cell is known as the tube cell. So pollen grain, 2 cell, one is generative, other is the tube cell. So generative cell will divide into 2 sperm. It will form 2 sperm. While the tube cell, it will form the pollen tube. And through this pollen tube, the 2 sperm will move and go to the ovary. So pollination will take place. Pollen grains are transferred to the stigma to a flower, often facilitated by pollinators like insect or wind. So through insect or wind, as the flowers are showing, pollination will take place. And embryo sac development, embryo sac formation, megaspore undergo mitosis to form embryo sac. So like microspore, megaspore will also go repetitive division and it will form a embryo sac. And this embryo sac typically consists 7 cells. Okay. In microspore, 2 cells only, generative and vegetative. Generative will give rise to 2 sperm and vegetative will give rise to a pollen tube. But here it is 7 cell, 8 nuclei. So there are 7 cells and 8 nuclei. So 1 egg cell is there, 2 synergid cells are there, 3 antipodal cells are there, and 1 central cell with 2 polar nuclei are there. So what embryo sac contain? 1 egg cell, 2 synergid, 3 antipodials, and 1 central cell with 2 polar nuclei. That is, it is called 7 cells with 8 nuclei. There are 7 cells and 8 nuclei. So remember the terms one X cell, two synergid cells, three antipodal cells, and one central cell with two polar nuclei. Because we need these terms in our next chapter, that is the reproduction in flowering plants. So these are the terms. It is 7 cells, 8 nuclei. So next fertilization will take place. Pollination and pollen tube growth. Upon landing on a compatible stigma, pollen grains germinate and grow a pollen tube through the style to the ovary. So I have already mentioned this, that there is pollen receptivity. So compatible pollens will germinate and it will form a pollen tube, and through that pollen tube the two sperm will come, and one will fertilize the egg to from the diploid zygote, while the other sperm will fuses with the two polar nuclei to form a triploid endosperm, which will nourish the developing embryo. So this is called double fertilization. Next is seed and fruit development. So seed formation will take place. The fertilized ovule develop into seed containing the embryo, which will develop into new sporophyte. The embryo will turn into the new plant, and the endosperm, which provide the nutrition. Next is fruit formation. The ovary will mature and develop into fruit, which protect the seeds and aids in their seed dispersal. Next is germination and sporophyte formation. So once favorable condition arises, this seed, when these seeds will germinate, the embryo will begin to grow, developing into a new sporophyte. And developing into mature plants, the young sporophyte plant grows and matures, eventually producing flowers, and cycle continues. This is all about the life cycle of the angiosperms. From here, you will have some idea so that in the next chapter, we can learn more efficiently. So I have already shown you this. Hey, spores, mitosis takes place, mega gametophyte, microgametophyte, then gametes will come, then fertilization will take place, zygote will form, again mitosis will take place, then sporophyte will take, from the sporophyte, meiosis will takes place, then again mitosis. This is the whole process, whole diagram. So in angiosperms, the alternation of generation involves a dominant sporophytic space where the plant's main structure are present, and a reduced gametophytic phase, which is dependent on the sporophyte. So gametophytic stage is dependent on the sporophyte, and gametophytic stage is not dominant. Here is also sporophytic stage is the dominant. This alternation ensures genetic variation and adaptation through sexual reproduction, while the development of seeds and fruit aids in the successful dispersion and survival of the species. So this gives genetic variation and adaptation through sexual reproduction. So this is the dominant phase. Here is also sporophytic phase, and the gametophytic phase is dependent on the sporophytic phase. So now the classification of angiosperms, only two is there. So basically there are many groups, but their major groups are monocots and the dicots, also known as eudicots. So what is monocots? The unique feature, it has single cotyledon. That is why it is called monocots, monocotyledons. So there is only single cotyledon. Monocots have one cotyledon, seed leaf in the seed. Only one cotyledon is there. Then the leaf venation is parallel. So the leaf typically have parallel veins. So it has parallel venation. Next is fibrous root system. They generally have fibrous root system with numerous thin roots. So there is only one main root, and with that numerous thin roots will arise. That is the fibrous root system. That is flower parts, floral organs, petals, sepals, and stamens are usually in multiple of 3. So these all are usually multiple of 3, like 3, 6, 9. This way you will find. Next is vascular tissue are scattered throughout the stem rather than arranged in a ring. It is scattered in the stem. Anatomy is scattered in the stem. Means xylem and phloem is scattered. It is not arranged systematically. That is the monocotyledons. So this is the key difference of monocot and dicot. You can say in monocot, single cotyledon. Dicot, there is 2 cotyledon. Then leaves are long, narrow leaf, parallel veins in the monocot. And in dicots, you see broad leaf network of veins. This is the leaf of the dicots. Then vascular abundance, you can see it is scattered here. But in dicot, it is arranged in a ring. You can see this. Next, this is a bit, hiding. Okay, I will explain. In monocot, you see that floral parts are multiple of 3. Like 3, 6, 9. But in dicot, the floral parts are multiple of 4 or 5. Like 4, 8, 12 or 5, 10, 15 this way. So this is the differentiation. Single cotyledon, 2 cotyledon, Leaves are parallel veins, and here it is network of veins, long narrow leaf, broad leaf, fascicular bundles scattered, fascicular bundles in a ring, and here floral parts multiple of 3, here floral parts multiple of 4 and 5. So this is the structure of bean. That is a dicot, this is the monocot. You can see it. The cotyledons have epicotyle, hypocotyle, radical cotyledons. So this is we will learn in the reproduction part. So this is the shape. So reproduction, so pollination, monocots can be pollinated by wind, insect or other animals. So example include grasses and orchids which have specialized mechanism for pollination. And seeds typically have a single cotyledon and the endosperm is retained in the mature seed to nourish the developing embryo. So it is retained in the mature embryo, the endosperm. Example is the grass includes crop like wheat, rice, and corn. They often have small, inconspicuous flowers and are adapted for wind pollination. While next example is the orchid, known for their integrate and diverse flower structures. Orchid have complex mechanism to attract specific pollinators. So example of monocots are grass, poachi, and orchids. That is the family from Orchidaceae. Next is the dicots, which is also known as eudicots. So unique features, of course, two cotyledons. Dicots have two cotyledons. Seed leaf is in the seed. Then the phoenician is net-like leaf phoenician. It has network of veins. Number 3, it is taproot system. Earlier in monocot, it is fibrous root system. So they generally have taproot system with a main root and a smaller lateral roots. So flower parts, floral organs are usually in multiple of 4 or 5, like 4, 8, 12, or in 5, 5, 10, 15. Then fascicular bundles, fascicular tissues are arranged in a ring within the stem. It is arranged in a ring, but monocot, it is scattered. So reproduction, dicots are often pollinated by a variety of agents, including insects, birds, and wind. They have evolved specific floral adaptation to attract pollinators. And seed development, seeds typically have two cotyledons, and the endosperm is often absorbed into the embryo during the seed development. So it is similar. And examples of these dicots are rose and the sunflower. So rose is a very common ornamental plants. The flower are very beautiful and it attracts, insects, and it is multiple of 5. Like there are 5 leaves or 10 or 15 petals like that. And sunflower known for its large composite flowers made up of many small florets. Sunflowers are often pollinated by bees or other insects. Okay, so this is all about the angiosperms we have discussed. So these are some MCQs with the answers. You can go through it. Okay, so we are done with the all this chapter. Okay, everything is done in this chapter, and from the next we will discuss the sexual reproduction of the flowering plants. Okay, students, got it? If you have any doubt, you can ask me. You can write it down, or you can ask me in the next class. From the next class, we will go through the next chapter, the reproduction in plants. Okay. There is a chapter left, that is morphology. It will teach by the other teacher, that is Dipashri ma'am. So this is what for today. Okay. So hello ma'am, are you there?
Yes,
Ma'am,
ma'am.
yes.
Yes,
Class is
ma'am.
over today. Thank you.
Yes, ma'am. Thank you, ma'am. Yes, students, thank you. Again, we will meet on Monday morning 5 to 7. Right? So, bye everyone. Tomorrow, don't forget to write the weekend exam. And
Okay,
please
ma'am.
do attend classes daily. Please take the classes seriously and attend classes daily.
Okay,
Okay?
ma'am.
Teachers are putting very much effort. మీకు రియలిస్టిక్ గా కూడా చూపిస్తున్నారు ఎట్లా ఏంటి అని చెప్పేసి. సో ప్లీజ్ షో సమ్ ఇంట్రెస్ట్. ఓకే, ప్లీజ్ అటెండ్ డైలీ. So bye everyone. We'll meet on Monday.